Welcome back to Peking University MOOC Bioinformatics Introduction and Methods. I am Ge Gao from the Center for Bioinformatics, Peking University. Let's continue this week's topic, sequence database search. Last time we have illustrated with an example on how to use BLAST to search databases for similar sequences of the query sequence within a selected species or across species. This can facilitate the study of the function and evolution of the query sequence. In this unit, we will introduce the ideas behind the BLAST algorithm. As mentioned before, BLAST was first published in 1990. According to Google Scholar, its two main papers have been cited nearly 50,000 times each. It is one of the most widely used and appreciated algorithms in bioinformatics. So far, there have been more than 30 different toolkits developed for BLAST. Many papers have been written on how to use BLAST. There are even books devoted to this algorithm. Due to time constraints, we cannot cover every single aspect of BLAST here today. We will only introduce its basic ideas and algorithms. Students who are interested can get further information from the reading section. As mentioned before, in dynamic programming, the key to reduce the computational time is to reduce the number of cells to fill in as much as possible. Specifically, we should try to compute only those cells in or surrounding the optimal alignment path. BLAST is an extension of this idea. Specifically, BLAST will first find highly similar small segments between the two sequences, called the seeds. These seeds will then be extended outward to construct the alignments. Finally, BLAST will compute the statistical significances for these alignments in order to reduce false positives. Specifically, BLAST will first slice the query sequence into multiple small segments called seed words. Then, BLAST will search the pre-built index table of all seed words in the sequences in the database. Then, BLAST will search a pre-built index table of all seed words in the sequences in the database to find the query sequences seed words which signifies candidates matches between the query sequence and sequences in the database. Students with background in computer sciences may realize immediately that it would take only linear time or even near constant time to search an index table. This will improve the computational efficiency considerably. By locating candidates for every seed, we can get a heat map between the query sequence and candidate match sequences in the database. As discussed in the previous unit, the optimal alignment path should be parallel to the main diagonal. Therefore, we can discard those isolated hits. We keep only the hit clusters, each of which has two or more successive hits parallel to the main diagonal. This will reduce the search space further. Then, for each hit cluster, we can extend outward from the two endpoints until the total score has decreased below a given value x. Then, for each hit cluster, we can extend outward from the two endpoints until the total score has decreased below a given value x. Next, we can identify the optimal alignment for each extended hit cluster by dynamic programming, thus reducing the computational time considerably. BLAST also uses the same other tricks to further enhance its speed and sensitivity. First, BLAST will mask, i.e. mark as to be ignored, low complexity sequences that have numerous copies in the genomes to avoid excessive false positive hits. Whether a sequence is low complexity is measured by its information content. This formula is very important. Without it, there are too many seed words to choose and use. More importantly, without it, it cannot avoid selecting seed words from the low complexity regions, leading to lots of false positive hits. For example, the CA-CA-CA repeat is a typical microsatellite sequence that has a K value of 0.36. This sequence will thus be masked to prevent it from giving false positives in database searches. Also, to improve the sensitivity, BLAST will consider not only the seed words from the query sequence, but also the neighborhood words similar to the seed words. The similarity here is evaluated by the substitution matrix. For example, the similarity between the seed word DKT and its neighborhood word DRT will be 6 plus 2 plus 5, which equals 13. Similarly, the similarity between all possible neighborhood words and a seed word can be worked out. Of course, we should only consider highly similar neighborhood words to reduce false positives. In practice, in the latest version of BLAST, a neighborhood word will only be considered if it has a similarity of 11 or more with the seed word. We will illustrate this concept now and demonstrate why it is essential. First, for a peptide sequence of length L, the probability of a random generated sequence being identical to this peptide is 1/20th to the power of L. If L is 6, then this probability is 1.56 times 10 to the power of -8. Now consider using this peptide sequence to search this SwissProt database. The expected number of perfect matches by chance will be 3.65. Whether or not there is no real match. Therefore, we need a method to objectively evaluate the statistical significance of an alignment. After we get an alignment, we need to assess its statistical significance to ensure that this alignment does not just arise by chance. BLAST uses the E-value to evaluate this significance. Briefly, E-value denotes the expected number of alignments that can happen by chance to have a score equal to or greater than that of the current alignment. Specifically, if the E-value of an alignment is 10, it means that 10 alignments are expected by chance to happen whose scores are equal to or greater than that of the current alignment. Please note that it is not a probability. It is an expectation and may be larger than 1 as defined above. The expectation is worked out from this formula. We will not discuss this formula in detail today, but I will explain every variable in this formula. The M is the length of the query sequence. The N is the size of the database. The E is the natural logarithm, and the S is the score of your alignment. The K and lambda are related to the scoring matrix, serving as the normalization factors. As in the formula below shows, the E-value is directly proportional to N, the size of the database. In other words, the bigger the database, the more likely you will get an alignment just by chance. This is consistent with the example we have just discussed. The e-value is also directly proportional to m, the length of the query sequence. This is because BLAST is a local alignment. It does not require the alignment to cover the full length of the query sequence. Intuitively, the e-value is negatively correlated with the score S. Alignments with higher scores are thus less likely to occur by chance. The lambda and k in the formula are two correction coefficients related to the scoring matrix and search space. The lambda and k in the formula are two correction coefficients related to the scoring system and search space. They are used to correct the effects of different scoring matrices and search spaces on the final results. To make it easier to understand, we can convert between p-values and e-values. As shown in the figure, the e-value and the p-value, i.e. the probability, are nearly the same when they are less than 0.1. In particular, when p is 0.05, the e-value is 0.0513. 0.05 is often used as a cutoff for e-value. The smaller the e-value, the more reliable the result is. Good. Now let's summarize. BLAST is heuristic, making it different from the previous dynamic programming-based algorithms. For example, Needleman, Wunsch, and Smith-Waterman. It cannot guarantee the optimal solution, but it will find a solution that is good enough in much less time. Specifically, BLAST uses a seeding and extending strategy and applies dynamic programming only in a limited number of short regions. This has reduced the computational time and improved the speed effectively. However, the reduction of computational time comes at the cost of a decrease in sensitivity. This is a common trade-off for all the heuristic algorithms that we will discuss in later courses. Good, we have briefly introduced the basic ideas behind the BLAST algorithm. Here are the summary questions for this unit. These questions are not part of the assignment. You are encouraged to think about them and discuss your answers and ideas in the online forum. That is all for this unit. Thank you.